Hello everyone, this is Lecture 1D, the final lecture for Week 1 of Applied Immunology. This lecture will introduce us to the organs of the immune system, which are called lymphoid organs, and represent major sites of immune cell development and activation. We'll also cover the lymphatic system, which is used in combination with cardiovascular circulation to transport immune cells around the body so that they can effectively patrol for signs of infection or injury. Lastly, we'll end with a discussion of where we connect the specialized anatomical structures of lymphoid organs to their essential functions in the generation of a successful immune response. We'll start off today by talking about sites of immune cell development and differentiation, which are referred to as lymphoid tissues or lymphoid organs. As we discuss these organs, I want you to keep this immune cell differentiation schematic in mind. So think about where stem cells and progenitor immune cells are generated, and where immune cells can then develop and undergo maturation within various tissues throughout the body. Additionally, to briefly cover some vocabulary terms, leukocyte refers to any type of immune cell, while lymphocyte refers primarily to adaptive T and B lymphocytes, and myeloid cell is used for most innate immune cells, which are derived from a common myeloid progenitor, or CMP. Now, lymphoid organs can be categorized as either primary or secondary lymphoid organs. Primary lymphoid organs are where all leukocytes originate from. And so, a major site of leukocyte development is the bone marrow, which generates common lymphocyte progenitors, or CLPs, as well as common myeloid progenitors, or CMPs. Notably, although both B and T cells are derived from CLPs in the bone marrow, T cells must travel to the thymus where they undergo an additional selection and maturation processes to complete their development, which we'll get into later. And the thymus is this butterfly-shaped organ located above the heart near the throat. In addition to primary lymphoid organs, there are a number of secondary lymphoid organs located around the body. These organs have high concentrations of T and B lymphocytes and are sites of adaptive immune activation through a process that we broadly refer to as immune priming. We'll focus today on two primary types of secondary lymphoid organs, the lymph nodes and the spleen. Lymph nodes are quite numerous and spread out all around the body, while the spleen is located on the left-hand side of the body just above the intestines. In addition to lymph nodes and spleen, there are a number of other specialized secondary lymphoid organs. Well, for the purposes of this course, we won't be covering them extensively, but just as a brief introduction, there are Peyer's patches, which line the small intestine and are important for immune priming against gastrointestinal infections, as well as the adenoids and tonsils, which are located in the head and neck area where they are important for other types of infections. The immune system needs to be able to respond to signs of infection or injury all around the body, so how do leukocytes actually get around? Many immune cells use cardiovascular circulation, which is an anatomical system that I'm sure everyone here is fairly familiar with. Immune cells also rely on a complex network of lymphatic vessels that are interconnected with lymph nodes to successfully travel through the body. Lymphatic vessels are just as numerous as blood vessels, while many different types of lymph nodes are positioned around the body and often named for their anatomical positioning. So mesenteric lymph nodes are found in the gut, popliteal lymph nodes at the back of the knee, auricular lymph nodes behind the ears, etc. And immune cells rely on both lymphatic and vascular vessels to efficiently circulate throughout the body. As everyone here is well aware of, or at least I hope you are, the cardiovascular system circulates blood throughout the body, while the lymphatic vessels are responsible for circulating lymph. And lymph is simply interstitial fluid that is pressed out of vascular capillaries, so as the diameter of blood vessels decreases, hydrostatic pressure increases, resulting in the egress of interstitial fluid that basically just bathes the cell in any given tissue or organ. This clear fluid is then absorbed by lymphatic capillaries, carrying immune cells such as antigen-presenting cells or APCs, as well as soluble particles such as antigens into the lymphatics. Non-lymphoid tissues, or NLTs, refer to any tissue that is not a primary or secondary lymphoid organ, and these are often the tissues where lymph enters the lymphatic system. In immunology, these are often referred to as peripheral tissues or simply the periphery. Lymph collects from non-lymphoid tissues and then drains to the closest lymph node, which is therefore referred to as the draining lymph node for a given tissue or organ site. For example, if you have a throat or ear infection, the auricular lymph nodes under the jaw would be considered the draining lymph node for that infected tissue. As we'll discuss in the next slide, these lymph nodes contain high concentrations of T and B lymphocytes that undergo activation required for the control of infection. And the activation of these cells in a draining lymph node is referred to as immune priming, which is associated with cellular proliferation and inflammation, explaining why you can often feel these swollen lymph nodes underneath your ears and jaw when you have something like a head cold. Now, once lymph arrives at a lymph node, it washes through a series of sinuses within the lymph node, then exits and eventually rejoins blood and circulation via the thoracic duct. As we discuss immune cell trafficking during the generation of immune responses, I want you to keep this circulatory pattern in mind. So fluid and cells generally travel through the blood and into peripheral tissues, where lymph then drains through the lymphatics into the draining lymph node before eventually rejoining blood via the thoracic duct. Secondary lymphoid organs are important sites for leukocyte interactions and immune priming, a term commonly used to refer to the initiation of adaptive immune responses. We'll primarily focus on lymph nodes and spleen when discussing secondary lymphoid organs. First, let's look at lymph nodes. So these monitor lymph for antigen, which can enter in the form of either soluble antigens or an antigen-presenting cell that is migrated from peripheral tissue to the lymph node while carrying antigen. Lymph arrives at the lymph node through the afferent vessel, which attaches to this outer region called the cortex. It then washes through a series of sinuses, first over lymphoid follicles that contain B cells. This is typically referred to as the B cell zone on the outside, and then over the paracortical area containing dense regions of T cells. And this is not surprisingly referred to as the T cell zone, which is a bit deeper inside of the lymph node. Lymph then exits through the efferent vessel on the inner curved edge, where it eventually re-enters vascular circulation. Next, we have the spleen. While the lymph nodes filter lymph, the spleen filters blood and surveys it for antigen and other signs of infection. Notably, the spleen also removes old red blood cells, or RBCs, using a specialized population of phagocytes called hemophagocytic macrophages. Blood enters the spleen through an artery, where it washes over lymphocyte-rich white pulp. And these follicles are structures that contain B cells, while the PALS region contains T cells. Spleens are divided into regions of red pulp and white pulp. As noted, white pulp contains high densities of lymphocytes, while red pulp contains blood and high concentrations of other innate immune cells such as monocytes, granulocytes, and red pulp macrophages. Once blood circulates throughout the spleen, it exits through a vein to re-enter vascular circulation. Now, although the spleen and lymph nodes differ with respect to the fluid compartments that they filter and their specific anatomical features, there are some very important similarities between these two organs. Both are centralized organs that monitor circulating fluids for signs of infection by receiving antigen that is brought in from peripheral tissues. Both secondary lymphoid organs house high concentrations of naive lymphocytes that can undergo activation and initiate an adaptive immune response. Again, this process is commonly referred to as immune priming. This organ architecture is especially important when considering that for any given antigen, the immune system needs to find a very small number of lymphocyte receptors that may bind to and recognize that antigen. Therefore, the high density of naive lymphocytes that are housed in secondary lymphoid organs helps to facilitate cellular interactions that allow rare cells to find each other. That is, it increases the likelihood of a given lymphocyte antigen receptor to find an APC that's presenting cognate antigen. In summary, today we've learned that immune cells originate in primary lymphoid organs from various leukocyte progenitor populations, and that these cells then circulate throughout the blood, peripheral tissues, and secondary lymphoid organs to patrol for and respond to immunological insults such as infection. We've learned that secondary lymphoid organs filter distinct circulatory compartments, so lymph nodes filter lymph while the spleen filters blood, in an effort to survey for signs of infection in peripheral tissues. We've also discussed how secondary lymphoid organs possess specialized anatomical features that allow rare cells to find each other. So the high concentrations of lymphocytes and lymph nodes in spleen are exposed to circulating fluids that can carry both soluble antigens as well as antigen-presenting cells, and that this increases the likelihood of lymphocyte antigen receptors encountering cognate antigen that they can bind to, which enables their activation. Lastly, we've learned that these interactions are essential for the successful activation of adaptive immunity by innate immune cells in a timely fashion. This concludes the lecture portion for week 1 of this course. As a quick reminder, you do have some assignments which accompany these lectures. One is a reading assignment to complete using the Immunobiology textbook by Janeway for this course, so please consult the syllabus for specific chapter and page ranges for that. Number 2 is that you have your first graded assignment for the course this week, which is your first discussion board on Canvas. But this is an easy one, though, because you just need to introduce yourself to the class. So please view the week 1 discussion board prompt on Canvas for some more information about that. Lastly, you have your first opportunity for extra credit by completing the reading quiz for this week. So this is just a reminder that this is an open book and open note quiz. It's only 5 questions. So go ahead and complete that if you'd like to start off the course with some extra credit points. Good luck with all of your assignments, and I'll see you all next time for week 2 of Applied Immunology.